Hi, everybody. Thanks for joining me on the Google Hangout today. My name is Heather Thorstensen. I'm the Manager of Communications for Sigma Xi, the Scientific Research Society. If you're not familiar with Sigma Xi, we are an interdisciplinary honor society for scientists and engineers. Today, I'm going to be speaking with a leader in vision research, Dr. David Williams. He is a professor of medical optics, the director of the Center for Visual Science, and dean for research in arts, science, and engineering at University of Rochester. He's getting other awards this year, but one of them is an award from Sigma Xi. He's going to be getting Sigma Xi's William Procter Prize for Scientific Achievement. This is an award that we give out to recognize a scientist's outstanding contribution to research. And also, the scientist has to have a demonstrated ability to communicate this research's significance with other scientists in different disciplines. So Dr. Williams will give a lecture and receive the award at Sigma Xi's annual meeting in October in Kansas City, Missouri. You can find more information about that meeting on Sigma Xi's website, which is sigmaxi.org. Today we have the ability to ask questions on the Hangout. So you should see on the right side of your screen a white column where you can type in your questions. If you don't see that, you can scroll over to the left side of your screen and look for the Q&A app. Click on that and hopefully that will bring up the questions for you. We're going to be taking questions toward the end of the Hangout. Dr. Williams, thanks for joining me.
Thank you, Heather, and thanks so much to your society for bestowing that wonderful award on me. I'm very honored and flattered to receive that.
Let's start by talking about some of the research that you've done and are doing. And one of the things we had talked about was how your research is incorporating some technology from the field of astronomy.
Well, that's right. So I'm a vision scientist, and throughout my entire career, really, I've been trying to understand how it is people can see. My research has always had a very strong optical component to it. I try to develop new optical technologies that allow us to study how people see, both in the case of people with normal vision and also people now with diseases of the eye. I try to use optics in novel ways in order to understand how the retina works and how it fails. And about 20 years ago, I had an interest in trying to separate the optical effects on vision from the neural effects. That is, your overall visual experience is governed both by the limitations set by the optical properties of your eye and also by the neural capabilities of your retina and brain. And I came across the idea that astronomers had developed that it was possible to correct aberrations in a novel way. And so when astronomers are trying to look up at the stars through their telescopes, they use a technology called adaptive optics to correct the optical defects of the eye. And what I realized was I could take that same technology, modify it slightly, and not correct the aberrations that occur in the atmosphere, which is what plagues ground-based astronomy, but instead correct aberrations in the eye with unprecedented accuracy. And so we figured out how to measure these aberrations very accurately, and we used this adaptive optics technology to correct these aberrations. And it allowed us to do two things. It allowed us to improve people's vision. If you were looking through a device, whether it be some glasses or contact lenses or a special instrument built to correct the aberrations we could measure, you could have improved eyesight. And then also it allowed us to look inside the eye with much greater clarity than we'd ever been able to do before. And now we can resolve single cells in the back of the eye with the special kinds of ophthalmoscopes that we've been developing over the last 2 decades now.
And so this is taking vision research in a whole new way. I've heard that it's gone from just looking at farsighted, nearsighted, and astigmatisms to much more than that. That's right.
That's right. So when, we're all familiar with spectacles, and, one of the most remarkable medical inventions ever. These things are great. They're wonderful if you're myopic, for example. These are just, or any sort of vision defect, these things are great. They correct the 3 most important optical defects in people's eyes. But what we showed is that with this technology we developed in the 90s, we could we could measure way, way more aberrations than just 3. We could measure dozens of aberrations, and all normal eyes have these aberrations. And then there are abnormal eyes that have even more aberrations, that have what we call higher order aberrations that that really, in some cases, for example, eyes that suffer from a disease called keratoconus, you're essentially legally blind unless you correct these aberrations. So we now had a tool that allowed us to make a much more complete description of all the different things that can go wrong with the eyes that will reduce the quality of the image available to the retina and the brain. So the first step in fixing that is measuring the aberrations more accurately than we ever could before. And we now have that tool in place, and you can now go buy one of these instruments because it's been commercialized. You can buy an instrument that will make these measurements for you. So once we had that ability, then, as I said before, we could go back and use it for 2 things. Either for light entering the eye, we can improve people's vision. For light coming out of the eye, we can take pictures. We can use that light to take pictures of the inside of the eye with a clarity and resolution that's really microscopic for the first time, and we can resolve single cells back there. Were never visible before in a living eye.
Could you explain how, when you look into the eye and you can see these aberrations, what is it that you're doing what is it that you have changed that is correcting the vision more?
What we have changed is we have interposed, in a particular, I'll tell you about several ways that we can make these improvements. You could do, for example, laser refractive surgery, and nowadays modern methods to do laser refractive surgery or LASIK employ the technology we developed in the 90s. That essentially everybody who has LASIK these days, whether they know it or not, they're using this wavefront sensor that we developed, or a variant of it, in order to improve the outcome in laser refractive surgery. So one of the things you can do is you can make a measurement of the aberrations and measure dozens of aberrations instead of just a couple. And then you can use that information to guide a laser that is sculpting your cornea in such a way as to correct the aberrations, those aberrations in your particular eye. And again, everybody has a different pattern of aberrations. It's like everybody has a different fingerprint. You have your own unique signature, which is that pattern of aberrations in each of your eyes. And so we need a special correction for every individual. And that laser refractive surgery procedure, which is sometimes called customized LASIK nowadays, is an example of a method that capitalizes on this ability to measure aberrations. On the other hand, if we're looking into the eye with our ophthalmoscope that's equipped with adaptive optics, we use a different method to correct the aberrations there. We use a mirror called a deformable mirror. Imagine a mirror surface that's flexible and has an array of little pistons on the back surface of the mirror that can push and pull on the mirror to create an arbitrary shape. What we can do then is we can use the aberration measurements to adjust the shape of that mirror in just the right way so as to correct the aberrations. Normally, you're used to using lenses like glasses use, or contact lenses, to correct aberrations, but you can also do it with mirrors. And we do it with this thing called a deformable mirror. Once you have a deformable mirror in your system, your instrument for taking pictures of the back of the eye, you can now collect a microscopic view, because you no longer have the blur that is normally present when you look into somebody's eyes. The blur that these aberrations normally produce, that blur is removed, and you have a really sharp microscopic view of the retina. The retina, of course, is composed of many, many different kinds of cells, the rods and cones everybody knows about, that are responsible for nighttime and daytime vision. And in addition, there are many other cells that are important in the retina and important to be able to see, both to understand how the retina works, and also if you're interested in developing novel ways of curing eye disease.
So in addition to enhancing LASIK surgery or improving LASIK surgery, is this research also being used to enhance like glasses and contact lenses?
Yes, just about every, contact lens design nowadays is in the companies that do that sort of thing they are using wavefront sensors to measure their contact lenses on the eye. So they know what aberrations the lenses are producing, and they know what they need to do to correct the shapes of those lenses to remove those aberrations. So it is true that this technology has improved the design of contact lenses, and it's also used for intraocular lenses. You know, when people get older and they develop cataracts are a clouding of the natural lens, and the lens has to be replaced with an artificial lens called an intraocular lens. And the design of those lenses is also being worked on and improved using the aberration measurements that we can make with this technology. So there's been widespread adoption of this method to provide this very complete description of the optical defects of the eye for a wide range of ophthalmic treatments, from contact lenses to intraocular lenses. Even spectacles to some extent are being improved. Spectacles are a little bit of a different case because your eyes move with respect to the spectacles, and that makes these higher order aberrations that I mentioned more difficult to correct with them. But still, there are applications of wavefront sensing to make sure that spectacles are performing the way they're intended to be designed.
And so what is the research that you're working on now?
Well, so what I'm doing now is we have been working for almost two decades now to develop improved cameras for taking pictures of the inside of the eye that use adaptive optics. And one of the really wonderful things from my perspective about this technology is how flexible it is, in the sense that you can combine adaptive optics with any other imaging modality that you might be interested in to get an even better instrument. So for example, it's possible to combine adaptive optics with a technology called optical coherence tomography, which so adaptive optics gives you better resolution in the retina. If you're looking at the retina and you're looking at single cells there, you have better resolution in X and Y. That is, if you're looking down on the retina, you can see single cells in that plane, in the plane of the retina, much better than without adaptive optics. But OCT, optical coherence tomography, gives you better resolution in the Z dimension, that is in depth. So by combining OCT with adaptive optics, you get an improvement in resolution in all three spatial dimensions. And that allows you to see the three-dimensional volume of the retina. The retina is not just a single surface, it's actually a multi-layered structure. And with adaptive optics combined with OCT, you have the ability to reconstruct the entire three-dimensional volume. So that's an example of how you can combine adaptive optics with another technology and get something even better. We've been, at Rochester, we've been combining adaptive optics with fluorescence imaging. Turns out a lot of structures in the back of the eye are intrinsically fluorescent, and in some cases we can use fluorescent dyes to make structures visible that wouldn't ordinarily be fluorescent. So we have the opportunity to study the retina in a novel way, and I'll give you one example. There's a new kind of microscopy called, relatively new, it's been available for the last 15 or 20 years as well, but it's called two-photon microscopy. And it's a kind of fluorescence imaging where you rely on two photons hitting a fluorescent molecule instead of one. And it has some very special properties. So we're using that technology. A colleague of mine, Jen Hunter, and I are working on that here at Rochester to observe the functional changes in the retina. It's not just about looking at the structure of the retina. We're also interested in trying to understand the function of the retina using imaging technologies. And two-photon imaging combined with adaptive optics gives us a microscopic view of function as well as structure. So that's exciting for us. We're able to see, for example, the metabolic pathway that allows the photopigment in the eye to be regenerated. So every time you open your eyes, and as long as you keep them open, light is entering your eyes, and it's being absorbed by molecules in the back of the retina called it's called rhodopsin. There's 4 different kinds of photopigments in your eye, and the rods and cones, and those are what you see with, right? And there has to be something, once a photopigment molecule has absorbed a photon of light, there has to be something that comes along and fixes that molecule again so it can absorb another photon, because it gets bleached by the first photon that hits it. So there's this regeneration method, or a cycle that occurs in the eye, and we're now able to actually observe that in a living eye. And this is very important because there are lots of eye diseases that impact that cycle and prevent it from working properly and ultimately lead to blindness. So we're able to now directly observe intermediate stages in that cycle that have never been seen before, in a living human eye at least, or a living primate eye.
So when you think about your research, what are your ultimate goals of all of this when you're trying to put it all together?
Well, these days, more and more, we continue to develop the technology. We're working on ways of, for example, stabilizing the image that you see. You know, one of the things about your eyes is that your eyes are always moving. Even if you try to hold them perfectly still, the eyes are always moving a little bit. And if you're trying to take a microscopic picture of the retina, you're looking at very, very tiny cells in the back of the eye. And you can imagine if the eye is moving, well, then you've got a moving target. So we and others, our collaborators around the country, have been working together over about a 10-year period to develop new methods to stabilize the image, so we can freeze that motion and capture even sharper images as a result. So we've been making improvements of that kind in the camera all along. But our ultimate goal now is very much more toward two kinds of things. One is we'd like to understand why it is that the retina is organized the way it is. The retina sends signals up to the brain via about 1.2 million fibers, 1.2 million neurons. These are the axons that make up the optic nerve. And that might sound like a big number, but 1.2 million is small when you think about the fact that you have, in a typical cell phone, maybe 10 million pixels in your cell phone. So how is it that the eye can get by and produce such a beautiful view of the world for us with such a small number of fibers? And furthermore, there are about 24 different kinds of fibers. And we don't know why the retina has to have so many different kinds of wires, if you will, to convey the retinal image to the brain. So one of our basic neuroscience goals is to try to understand why the retina is organized the way it is. And the more we learn about the retina, the more we appreciate that it's really a very smart, intelligent microprocessor. It is, after all, a part of the brain. And it is doing a lot of important things in processing the retinal image so that the brain can have something already pre-processed that it can interpret. So the more we study the retina, the more we realize how smart it really is. The second aspect of our research program that we're very much invested in now, especially going forward, we're going to be working much more closely on using this technology to improve cures for eye disease. One of the frustrating things about ophthalmology, as advanced discipline as it is that so often patients will come into an ophthalmologist's office and the ophthalmologist will be able to diagnose that disease really, really accurately. They'll say, you have macular degeneration of the dry form, and guess what? You know, we don't have anything we can do about it for you. We can tell you how to change your lifestyle to deal with the disease, but we can't cure it. And now there are, thanks to molecular biology, there are a host of new technologies on the horizon that are not yet ready for human use, but that are under development that I think in the next decade or two will produce major inroads in our ability to cure blinding eye disease, especially diseases that affect the outer retina, that is the rods and cones, diseases that damage the rods and cones, like macular degeneration. So I'm really excited about the future, and we're positioning our adaptive optics camera to accelerate the development of those technologies. These diseases kill cells, and what you need to be able to do if you're going to rapidly determine what works and doesn't work in the way of therapy, what you're going to need to do is view those cells in living eyes. It's one thing to have a model of this disease in an animal and track the and look at the animal's retina in a microscope after you've removed it from the eye, and then of course dozens, if not hundreds, of animals have to be used to develop a therapy for disease. It's another thing to be able to look into an eye, a living eye, and track the progression of that disease and the efficacy of the therapy that you have engineered to try to cure that disease. To be able to track that in real time in a single individual is a huge benefit. So that's what I'm really excited about, and I have a partnership now with 3 other institutions, investigator at Harvard and University of Wisconsin, and also in Basel, Switzerland. And we're working on 3 different approaches to curing blinding eye disease of the retina with them using our high-resolution camera.
Speaking of animals, one of the topics we thought we'd cover because we thought it would be interesting for our audience is the difference between what animals like dogs, cats can see versus humans. Can you talk a little bit about that?
Yeah, so we maybe a quarter century ago, we really didn't have a very complete understanding of what limits how well different animals can see. But nowadays, we do have a relatively complete theory. Thanks to a lot of theoretical work and experimental work, we now understand what the interplay is between the optical properties of the eye on the one hand and the neural properties of the retina and the brain on the other. And we can see that, for example, a fly has a visual acuity. It turns out, believe it or not, you can measure the visual acuity of a fly. You have to be very clever because they don't tell you whether they see the eye chart, obviously. But there are ways, believe it or not, that you can test behaviorally what a fly can and cannot see. And in these clever experiments that were actually done by Germans many years ago, nearly a century ago now, these experiments started there. And we now know that a fly is about 100 times worse in its visual acuity than a human. And a cat would be about 10 times worse than a human. So they have a much fuzzier view of the world than we have. On the other hand, we shouldn't get too smug about all this because there are animals that have superior visual acuity. The eagle is an example. Everybody talks about eagle vision, and yes, it's true. An eagle has a visual acuity that's about 2.5 times better than humans for 2 reasons. One, they have much better quality optics. The optical quality of the retinal image is better in an eagle eye than it is in a human. And also, they have a much finer grained retina than we have. So those 2 things together seem to be all you need to explain the visual acuity of the eagle. There are big differences too in terms of color vision. So dogs are colorblind. They do see color. They're not completely colorblind, but they only see probably what we would call blues and yellows, whereas we have a red-green color vision that they lack. But then there are animals such as pigeons that probably have superior color vision to humans. So again, we're not special in the animal kingdom.
And I want to remind everybody that we are going to be asking for questions in just a little bit. We have 5 minutes left, so not that much time. If you do have a question now, go ahead and type it into the right side of your screen. If you don't see a place to type in the questions, hover over on the left side and look for the icon that says Q&A on it. And while we're waiting for everybody to type in their questions, I'll ask the final question I had, which is what's coming in the future for human vision?
Well, I think that there are some exciting things on the horizon. I'm excited about these potential cures for eye disease that might involve, for example, the use of stem cells. You implant cells into the retina that you pre-program to differentiate into the kinds of neurons you've lost in the retina. That's an exciting possibility that might cure eye disease. There are even ways now of, through genetic engineering, take neurons that survive in the retina. This is an alternative approach to curing blindness. And that is to take neurons that are surviving in the retina, and even though those neurons might not have evolved to be light sensitive, it's actually possible now to insert special molecules using genetic engineering into the eye that make neurons that weren't light sensitive to light again. And so that's another interesting possibility that may eventually cure blindness. And finally, in terms of the refractive state of the eye, that is the optical properties of the eye, one thing that I'm very excited about is some of my colleagues here at the University of Rochester, Wayne Knox and Crystal Huxland, are working on a way of doing laser refractive surgery without the need to remove tissue. So if you have gone under, if you've had LASIK on your eyes, or if you're planning on doing LASIK, what they will do now using current technology is they'll use a laser to remove little bits of tissue and very tiny microscopic blasts of laser light that that vaporize tissue so that you can change the shape of the cornea in just the right way to correct aberrations. But what Wayne Knox and Crystal Huxland at Rochester have shown is it's actually possible to do this without removing tissue. And that's very exciting because it means you wouldn't have to, there's also a cut that needs to be made in the cornea for the current procedure to be performed, and that could be avoided. So it could be a much improved way of improving vision, and it would not require contact lenses or spectacles. Those could be part of the past if we could get this new technology fully developed. It's been demonstrated successfully in cats so far, but has yet to be developed for human use. But it's a very promising technology that may eventually remove the need for glasses.
Okay, I don't see any questions from the audience, so I'll ask another question, which is can you say how far away we are to what you were saying about your research helping to avoid blindness due to eye disease? Is there a time frame for that?
Well, I'd say that a lot of these technologies that I'm thinking about, the stem cells and what we call optogenetics, this restoring vision through genetic engineering, and these technologies will take some time. And one of our goals is to make sure they're thoroughly developed and tested before we deploy them on humans. It's eyesight is so precious that if you have any remaining sight, you certainly don't want to put that at risk trying some therapy that hasn't been thoroughly tested and explored in advance. So I think it's going to be probably another decade, I would say, before these technologies are available. So if you're young now and you have a grandmother who's got macular degeneration, I think we probably won't be able to help your grandmother, but we will be able to help you. And I think, and I hope. I'm not making any promises, but I'm very excited about what my colleagues are developing in terms of cures for diseases that are as devastating as that one and others like it.
And since we have technically one more minute, I also wanted to talk with you about how you're pulling in a multidisciplinary team at your center, because Sigma Xi is really into promoting multidisciplinary science. So I wanted to hear about all the different disciplines that you're working with.
Well, this is has been one of the most exciting things for me in developing this technology, is that it's required bringing together. It's all about people, really. It's not about technology in the end. And I've got this wonderful laboratory that brings together engineers. So the Institute of Optics here at Rochester has wonderfully trained graduate students who are optical engineers. Those people need to work side by side with scientists who understand the cell biology of the retina and how neurons work and how to record from neurons and all sorts of issues about how the eye works and what is the science behind vision. And then we also have medical people who understand disease, and they have to interface flawlessly with the engineers and the scientists. And finally, we're working closely with industry. So we have a couple of engineers from Canon, the camera company, who are embedded in our laboratory now, working closely with us to teach us about how to translate this technology into working commercial instruments. So it's really 4 different cultures all blended together in one laboratory. And that's what's required, really, to move technology from an idea to a working instrument that really will benefit many, many people worldwide, hopefully, in the relatively near future.
Great. Well, Dr. Williams, thank you for speaking with me today.
It's been a real pleasure, Heather. And again, thanks so much to your society for this wonderful award. Appreciate it.
Thank you. Bye.
Bye.